Good morning. This video is going to quickly explain what you're going to do for the protein synthesis activity. So at this point, you should have watched the video going over the steps of protein synthesis and completed the notes sheet. And now you will see that there is an activity where you're going to apply those concepts. So here's the sheet, the protein synthesis activity that you should have in that packet of information. It should be right following the end of the notes sheet. And so with an understanding of replication, transcription, and translations, you are ready to play the mutation game, which will help illustrate possible consequences of mutations. So read the directions provided, write the responses in the space provided, and use the amino acid chart and Table 2 mutation game genetic code as a reference chart for the translation step. So I'm going to just kind of explain this using number 1, and then you'll be able to apply the same concepts going through and finishing up the rest of the situation or the rest of the questions. So here is a normal non-mutated gene. So the DNA strand is listed right here. So you have the TAC, the TGG, the GTA. So this is the DNA strand that you're going to be starting with. So what you're going to do is that the first thing that you're going to do is you're going to translate this into, or transcribe it, I should say, into a messenger RNA. So you're going to take this DNA strand and you're going to make a messenger RNA. Now it says use the long paper labeled. If you are doing this in a face-to-face lab, there's long paper for you to complete this as a group. If you're doing this in the online lab, then you can just write it right on here. But you're going to transcribe this into a messenger RNA. Okay, so again, use your notes. So the T will become an A, the A will become a U, the C will become a G, and you're going to continue down that way. Then it says, number 1, put a square around a codon. So remember, a codon is a series of 3 nitrogen bases or 3 letters on the messenger RNA. Then you're going to translate that messenger RNA into a chain of amino acids. So you're going to have to draw in the transfer RNA, just like we did in the notes sheet. And then you're going to put the amino acid in the top part, and then you're going to lock it into place using the anticodon. Okay, so you're going to have to use the amino acid chart to decode those codons. And I included the amino acid chart on here for you. So here's that amino acid chart. So again, remember, you're going to look at the first base here, the second base along the side, and then the third base along this side in order to figure out what the codon is coding for, which amino acid. So you're going to put all those amino acids back into a line. So for number 2, you'll use that chart. Then it asks you to circle an anticodon. So remember, an anticodon is a group of 3 nitrogen bases that's found on the transfer RNA. Label which step is transcription and which step is translation. So transcription is going from a DNA to the messenger RNA. Translation is going from a messenger RNA to an amino acid or to a protein. And then technically at this point, the protein product is done because you'll have a chain of amino acids. But in order to kind of help visualize this process, what I want you to do is I want you to use the codons from the messenger RNA strand, and you're going to plug those into table 2. So you're going to be using the other table that's at the end of this document in order to find the codon that's going to give you a specific letter. And your codons then will actually spell a word. So for this first set of 3 letters, nitrogen bases, so TAC, so on the messenger RNA, that would be an AUG. Right, so that's the codon. So if you go and you're going to use the table that I just showed you to figure out what amino acid it is, and then what you're going to do is you're going to go back. So this table will tell you the amino acids. So AUG is going to be a methionine, so that's what you would answer for the first part. But then you're going to take that AUG for that last step, and you're going to find it on here. So AUG, find the A, the U, and the G, which is right here. And it says start. So you would just write start in there. So then when you find the next codon, it was going to give you a letter. So, and the letters, once you put all the codons into place, is going to spell a protein, a product. So that's what you're going to do for these. And you're going to do the same things for questions 2 and questions 3, except you're going to be dealing with a mutation. So you're kind of going to have to compare these DNA strands to the original one, figure out what kind of mutation it is, look to see what kind of product is made, and hopefully you're going to be able to see how mutations can change the different gene products. If you have questions about any of this, advocate for yourself and get a hold of me. Email, phone call, text message, we can Zoom, and I will help you with this. Have a great day.